Glycolysis. Okay, this is very important topic. What is glycolysis? We all know glycolysis is also called Embden glycolysis. Another name is Embden-Meyerhof pathway. Embden-Meyerhof pathway. Glycolysis, it is given in your handouts. I again say, if you don't remember the each and every step very sequentially, it's okay. But I will say that each and every step of glycolysis is important. Question is asked. So let me start with glucose. Glucose is converted to glucose 6-phosphate catalyzed by hexokinase. Catalyzed by which enzyme? Hexokinase. Okay. Glucose, how many carbon atoms are there? 6 carbon atoms are there. In glycolysis, end product is pyruvate or lactate. Pyruvate, 2 molecules of pyruvate are produced. So you can say pyruvate must contain how many carbon? 3 carbon. Pyruvate must contain 3 carbon. Lactate must contain 3 carbon. Yes, let me try to show you. Yes, I have tried to make some animation. Look at the screen. This is glucose. If I say this is the cyclic structure of glucose, yes or no? Yes, this is cyclic structure of glucose, and this is ATP, means AMP, 2-phosphate, this is ATP, AMP plus phosphate, ATP, adenosine triphosphate. This is hexokinase or glucokinase. What is the function of this hexokinase or glucokinase? Look at the screen. Hexokinase will come and will take phosphate. From ATP and will give to glucose. And when this phosphate, it will join phosphate from ATP to glucose. Yes, it will transfer phosphate from ATP to glucose. Glucose will be converted to glucose 6-phosphate. Yes or no? Yes. So I say that hexokinase, what is the function of hexokinase? You can say hexokinase transfer a phosphate group from ATP to glucose. When ATP to glucose, ATP is converted to ADP and glucose is converted to glucose 6-phosphate. So we say that hexokinase transfer a phosphate group, and any enzyme which transfer a phosphate group, that is classified as transferase. Classified as transferase. Yes, I will discuss so many enzymes. You know that there are thousands of enzymes in our body. Yes, there are thousands of enzymes in our body. These enzymes are classified into 7 classes. Previously it was 6, now these are 7 classes. So I will write in the corner. Yes, you will write first class number 1 is oxidoreductase. Class number 2 is transferase. Class number 3 is hydrolase. Class number 4 is lyase. So you can easily remember, in any reaction, if oxidation or reduction is occurring, you will say that enzyme belong to oxidoreductase. Means any enzyme catalyze a reaction in which oxidation and reduction is going on, that is called oxidoreductase. Okay, class 1 is oxidoreductase. Class 2 is transferase. Transfer occur between two things. Yes, I just told you transfer occur between two things. Any enzyme which transfer a group is classified as transferase, and transfer occur in between two things. That's why EC number 2. Yes, they are given classes or enzyme commission number. If I say that oxidoreductase is class 1, or you can say oxidoreductase, the enzyme commission number is 1. Question is asked, what is the enzyme commission number? of oxidase 1. What is the enzyme commission number of 2? Transferase 2. Hydrolase cut a bond with addition of water. So HOH, water. How many letters are there? 3. So hydrolase is EC number 3. Lyase, Lyase, lyase. Y looks like 4. So yes, 4. Lyase is 4. EC number 4. Isomerase, EC number 5. I write 5 like this and I say isomerase, EC number 5. 6. Ligase, gay. Ligase, gay. Gay means chakka, sixer. So sixer, yes, in Hindi sixer, in Hindi chakka, sixer, gay. So one student told this mnemonic. 7, that is translocase. So you will remember the class, and if I ask you that hexokinase is classified as transferase, so any enzyme which transfer a group is classified as transferase. So hexokinase is transferase. It transfer one phosphate from ATP to glucose, that's why it is transferase. Now you will write in the right corner again, all kinases are transferase. First question. Sec means hexokinase, phosphatidylkinase, glucokinase, pyrophosphoglycerate kinase. All kinases are transferase. All kinases are transferase. All kinases require magnesium as cofactor. This is frequently asked question. All kinases require magnesium as. All kinases are transferase. All kinases require magnesium as cofactor. Third thing, if kinase is used, either ATP is used or produced. If kinase is used, or better, if you want to avoid confusion, if kinase is there, either in any reaction, if you find kinase, either ATP is used or produced. In any reaction, if you find kinase, either ATP is used or produced. And fourth point, if kinase produce ATP, if kinase produce ATP, that is known as substrate level phosphorylation. Substrate level phosphorylation. Substrate level phosphorylation. So this is, now look at this. Glycolysis, Hampton-Meyerhof pathway. First, glucose is converted to glucose 6-phosphate, catalyzed by hexokinase. All kinases are transferase, require magnesium as cofactor. So cofactor of hexokinase is magnesium. In this, ATP is used or produced. In this, ATP is used. Yes, got the point? Glucose 6-phosphate and fructose 6-phosphate are isomer. Glucose and fructose are functional isomer. So name of enzyme is isomerase. EC number of isomerase, please do reply. What is the EC number of isomerase? 5. Okay. Fructose 6-phosphate is converted to fructose 1,6 biphosphate. Good, Usha. Fructose 6-phosphate is converted to fructose 1,6 biphosphate by phosphofructokinase. Phosphofructokinase. Okay, all kinases are transferase. So phosphofructokinase is transferase, require magnesium as cofactor. Either ATP is used or produced. In this, ATP is used. So phosphofructokinase, again magnesium is cofactor and ATP is used. Clear? And fructose 1,6 biphosphate is produced. Clear? Now, fructose 1,6 biphosphate is having 6 carbon, glucose is having 6 carbon, so it is also having 6 carbon but 2 phosphate. It is converted to 2 molecules. Of one molecule is glyceraldehyde 3-phosphate having 3 carbon, DHAP having 3 carbon, means one compound is converted to two. That's why the name of enzyme mnemonic aldolase. Aldolase. एक के दो. In Hindi we say एक के दो. Aldolase. Aldolase, sometimes we say simply aldolase, sometimes we say aldolase A. Aldolase A. Clear? Aldolase or aldolase A. So glyceraldehyde 3-phosphate and DHAP. If you see the structure of glyceraldehyde 3-phosphate and DHAP, you will find they are isomers. No need to remember the structure, no question till now, but you must have an idea. Glyceraldehyde 3-phosphate and DHAP are isomers. Yes, they are isomers. That's why the name of enzyme would be isomerase. Yes, the name of enzyme would be isomerase. Now, this glyceraldehyde 3-phosphate, you can say DHAP. When DHAP is converted to glyceraldehyde 3-phosphate, suppose I say this reaction is occurring in this direction. Okay, means DHAP is converted to glyceraldehyde 3-phosphate. So you can say that in glycolysis you produce two molecules of glyceraldehyde 3-phosphate. Now glyceraldehyde 3-phosphate, why I have given handouts, you will not waste your time. Although I usually say if you have written the compound yourself, it would give a better memory. But dear friends, you have read these things so many times, but they are so close. Their words, their names are so close that they produce interference. What I said, they produce interference. So you will not try to remember the exact word. You will remember the step by special features. Just like I say phosphofructokinase, all kinases are transferase. So in each two kinases we used, and these are isomer, that's why isomerase. When question would be asked, even if you don't remember the steps, you will be able to solve question if you have read in this way. Okay, now fructose 1,6 biphosphate, 6 carbon, 2 molecules of 3 carbon, they are isomers. That's why the enzyme is isomerase, and DHAP is coming to glyceraldehyde, so 2 molecules of glyceraldehyde 3-phosphate. Next enzyme is glyceraldehyde 3-phosphate dehydrogenase. Now I would say in complete biochemistry, whenever you find the word dehydrogenase means removal of hydrogen. Hydrogen will be taken up either by NAD or FAD or NADP. If it is taken up by NAD, it forms NADH. If it is taken by FAD, it forms FADH. If it is taken by NADP, it forms NADPH. NADH produce 2.5 ATP, FADH produce 1.5 ATP, NADPH produce no ATP. So in complete biochemistry, wherever you find the word dehydrogenase means removal of hydrogen. Hydrogen will be taken up either by NAD, FAD or NADP. Most commonly NAD to form NADH, produce 2.5 ATP. Okay, NADPH, no ATP. I told you the function of NADPH is reduction, no ATP, 0 ATP. Okay, so we say that glyceraldehyde 3-phosphate dehydrogenase means hydrogen is removed, it is taken up by NAD, because 2 molecules of glyceraldehyde 3-phosphates are there. So I can say 2 NAD will be converted to 2 NADH, and this glyceraldehyde 3-phosphate is converted to 1,3-biphosphoglycerate. 1,3-biphosphoglycerate. Yes, so in this there was one phosphate. Here there are two phosphate. So one phosphate comes from inorganic phosphate. This question has been asked in NB. Yes, not a good question, but they have asked that the second phosphate comes from in this reaction when glyceraldehyde 3-phosphate dehydrogenase is working. It take one inorganic phosphate and attach to this to make. So out of these two phosphate, one phosphate is inorganic phosphate. This is the only reaction in which inorganic phosphate join to the substrate like this. Clear? So question has been asked that this second phosphate comes from ATP or inorganic phosphate. Yes, most of time, yes, you have seen this phosphate come from ATP. This phosphate come from ATP, but in this phosphate come from inorganic. Yes, now 1,3-biphosphoglycerate is converted to 3-phosphoglycerate by phosphoglycerate kinase. All kinases are transferase. So phosphoglycerate kinase is transferase. All kinase require magnesium as cofactor. So phosphoglycerate kinase will also require magnesium as cofactor. Either ATP used or produced in this, ADP is converted to ATP. So ATP is produced. And I told you if kinase produce ATP, I told you if kinase produce ATP, that is called substrate level phosphorylation. If kinase produce ATP, that is called substrate level phosphorylation, because two molecules of 1,3-biphosphogyrates are there. So 2 ADP are converted to 2 ATP. So phosphoglycerate kinase produce ATP, and any kinase produce ATP, that is called substrate level phosphorylation. If you see, these two reactions are catalyzed by kinase, are irreversible. This is irreversible. Okay, means most of the kinase catalyze irreversible reaction. Again I repeat, most of the kinase catalyze irreversible reaction, but phosphoglycerate kinase catalyze reversible. So this is catching point. Question is asked. All kinase catalyze irreversible reaction, but phosphoglycerate kinase catalyze reversible. Now 3-phosphoglycerate is converted to 2-phosphoglycerate by enzyme mutase. By enzyme mutase. What is the name of enzyme? Mutase. Mutase is an, yes, mutase. What is the function of mutase? Yes, let me show you what is the function of mutase. Mutase, yes, what is mutase? Yes, I told you 3-phosphoglycerate. Suppose this is, yes, suppose this is 3-phospho. Yes, phosphate is present at third position. Now mutase came. It took this phosphate and transferred to C2. So mutase transfer a phosphate group from C3 to C2, and any enzyme which transfer a group is classified as transferase, but it's not transferase. But it is not a transferase because it transfer a group within a molecule. So first question, definition of question, mutase transfer a group, yes, within a group. What I said, mutase, yes, mutase transfer a group within a molecule. Yes, transfer a group within a molecule. So mutase transfer a group within a molecule. That's why you can say molecular formula is same. If molecular formula is same, they are isomer. So mutase is not transferase, it is isomerase. It is isomerase, not transferase. So previously 15-20 years back, mutase was classified as transferase, but now long time back it was changed. Nowadays, no controversy, mutase is an isomerase. Mutase is an isomerase. Definition question is asked: transfer a group within a molecule. And you all, yes, this is also reversible. This is also reversible. 2-phosphoglycerate is converted to phosphoenol pyruvate by enzyme enolase. By enzyme enolase. Okay, by enzyme enolase. Now phosphoenol pyruvate is converted to pyruvate by pyruvate kinase. All kinases are transferase, require magnesium as cofactor. Either ATP is used or produced. In this, ATP is produced, and if kinase produce ATP, that is called substrate level phosphorylation. Two molecules of 2-phosphoenol pyruvates are there, so 2 ADPs are converted to 2 ATP. Okay, and pyruvate kinase, yes, the cofactor for pyruvate kinase is potassium, then magnesium. Yes, I have told you all kinase require magnesium as cofactor. Pyruvate kinase also require magnesium as cofactor. But if question is asked which is better answer, potassium would be the better answer for cofactor of pyruvate kinase. So I have told you this glycolysis. In this glycolysis, you can say there are 3 irreversible steps. How many irreversible steps? 3 irreversible steps. One is catalyzed by hexokinase, second is catalyzed by phosphatokinase, third is pyruvate kinase. 3 irreversible steps are there, and these 3 irreversible steps are called regulatory steps. What I said? 3 irreversible steps are called 3 irreversible steps are regulatory steps. 3 irreversible steps are regulatory steps. Okay, regulatory steps. There is a difference between regulatory and rate limiting step, but rate limiting step is catalyzed by phosphofructokinase. Rate limiting step is catalyzed by phosphofructokinase. Okay, I know this has been little bit hectic, but yes, each and every step is question. Let me repeat it fast. We started with glucose glycolysis. When we started glycolysis, we know that each and every cell utilizes glucose for the production of energy, and glucose is the main source of energy. We know that glucose is undergoing glycolysis. Glycolysis means conversion of 6-carbon compound glucose to 2 molecules of pyruvate having 3 carbon. So glycolysis is glucose to pyruvate 3 carbon or lactate in anaerobic condition. We know this is a very simple thing. So glycolysis, what is the purpose of glycolysis? The purpose of glycolysis to produce energy. So we started glycolysis to produce energy. We started with hexokinase. ATP is used. 1 ATP is used here. 1 ATP is used here. So up to this step, you utilized how many ATPs? 2 ATPs. But you started glycolysis to produce energy, but you are utilizing. This is the rule. Whenever you want to produce large amount of energy, initially you must invest. What I said, whenever you want to produce large amount of energy, initially you must have invest. Rate limiting enzyme. Okay, yes. So we said that initially we utilize energy, means glycolysis, just like you are preparing, you have invested. In the same way, glucose is investing. That's why it is called preparatory phase of glycolysis. Up to this point, we say that preparatory phase of glycolysis. Okay, no mnemonic is required. First of all, glucose, energy, we put energy we want to add energy, that is kinase, 1 ATP used. Glucose is converted to fructose 6-phosphate, then again kinase 1 ATP is used. Now we want to break it, aldolase, एक के दो बना दिए, yes, glyceraldehyde 3-phosphate and DHAP, both are same, yes, both are having same molecular formula, that's why isomerase. Then the next enzyme is dehydrogenase. Whenever dehydrogenase is there, removal of hydrogen is taken up either by NAD, FAD or NADP, most commonly NAD to form NADH. Yes, NADH enter the mitochondria to produce 2.5 ATP by oxidative phosphorylation, we all know. 1,3-biphosphoglysrate is converted to 3-phosphoglycerate by phosphoglycerate kinase. All kinases are transferase, require magnesium as cofactor. This is reversible, and in this ATP is produced. If kinase produce ATP, that is called substrate level phosphorylation. 3-phosphoglycerate is converted to 2-phosphoglycerate. Yes, means phosphate is transferred from third position to second position. If any enzyme transfer a group, that is called, yes, within a molecule, that is called mutase. Question has been asked. Mutase is classified as isomerase. 2-phosphoglycerate is converted to phosphoryl pyruvate by enolase. Okay, now phosphoryl pyruvate is converted to pyruvate by pyruvate kinase. You will remember all these steps because each and every step has clinical significance. I will discuss one by one. So rate limiting enzyme is phosphofructokinase. Clear? Now one by one I discuss. Enolase, yes, if you have space here, you will write enolase is inhibited by sodium fluoride. What is the clinical question or clinical importance? Sodium fluoride or fluoride? Yes, means if you want to take blood sample for blood glucose estimation, yes, blood sample for glucose estimation, you know that blood is having RBCs, RBC utilize glucose, yes, RBC utilize glucose. Most of the cells in our body utilize glucose, yes. We added sodium fluoride, in this test tube. Why? Because we know that RBC utilize glucose. You took blood sample for glucose estimation. In this sample, sugar is there, RBC also there. RBC will utilize some amount of glucose if we don't add sodium fluoride. If I don't add sodium fluoride, how question was asked? There was an intern who took sample for blood glucose estimation. He forgot to take the tube having sodium fluoride, means sodium fluoride was not there in the test tube. He took the sample. Yes, he took sample and he deposited this tube, this test tube, next day in the lab. Blood glucose level was measured, and we found that in this test tube the result would be the estimated blood glucose would be option A falsely high, option B correct, C falsely low, because RBC will utilize some amount of glucose. Means if you take sample, always add sodium fluoride. Okay, so one clinical importance. Same clinical importance, you know that whenever we don't do brush, some food particles are there, glucose is there, carbohydrate is there, bacteria ferment on them. What I said? Bacteria ferment on them and they produce pyruvic acid, lactic acid. Whenever fermentation is there, glucose is converted to lactate. Yes, I am going to calculate the number of ATPs. Yes, 5 ATP will be produced. Pyruvate in anaerobic condition, pyruvate is converted to lactate, lactic acid. So in mouth, bacteria ferment the glucose and produce acid. When acid is there, we all know that acid is responsible for dental caries. That's why toothpaste, yes, we add fluoride. We add fluoride in toothpaste. क्या आपकी टूथपेस्ट में नमक है? क्या आपके टूथपेस्ट में फ्लोराइड है? Yes, so that is sodium fluoride. So fluoride we add in toothpaste because we know that dental caries are due to sodium, yes, due to acid, and this acid means fermentation of glucose produce pyruvic acid, lactic acid mainly, and if we inhibit this, that yes, this enolase, then there would be no lactic acid formation. That's why toothpaste are having fluoride. Now pyruvate kinase deficiency. Pyruvate kinase deficiency in RBC. I draw like this. In RBC there is sodium potassium ATPase. Sodium potassium ATPase. Okay, 3 sodium out, 2 potassium in. Okay, 3 sodium out, 2 potassium in, ATP is used. In RBC, glycolysis occur. Glucose is converted to pyruvate, then lactate. In these steps, one enzyme is pyruvate kinase. We found, yes, you all know, in RBC glucose is converted to pyruvate to lactate, or you can say the glycolysis occur, and one enzyme of glycolysis is pyruvate kinase. If pyruvate kinase is deficient in RBC, then ATP will not be produced, energy will not be produced, energy not produced. If energy not produced, then this ATP will not be there. This sodium potassium pump will not work. Sodium will accumulate. Sodium will not come out of this. Sodium will accumulate here. Okay. Osmosis. Yes, swelling of RBC. Hemolysis. Hemolysis. Yes, hemolytic anemia. Question is asked, most common cause of hemolysis, most common enzyme deficiency leading to hemolysis. Yes, answer is glucose 6-phosphate dehydrogenase. Question. First question, most common enzyme deficiency leading to hemolysis, G6P deficiency. Second most common enzyme deficiency leading to hemolysis is pyruvate kinase. Are you getting this? Are you getting this? So most common enzyme deficiency, again I say, if energy is not there in glycolysis, pyruvate kinase deficiency is there, glycolysis would not be there, ATP will not be produced, yes, sodium will not come out of the cell, sodium will accumulate, it will produce osmosis, water will enter, this RBC will swell up and will be hemolysis. There would be hemolysis. Clear? So question is asked, most common enzyme deficiency leading to hemolysis is G6PD. Second most common enzyme deficiency leading to hemolysis is pyruvate kinase. A 33, it is not 3, it is 33. 33-year-old man presented with symptom of weakness, fatigue, shortness of breath. Up to this, you can't say any diagnosis. In clinical practice, most of the patient, you will find her second, third patient would come, doctor साहब weakness is there, fatigue is there, shortness of breath there, चक्कर से आ रहे हैं, कमजोरी आ गई. yes, weakness hai, but his hemoglobin was 7. Now you can say all these symptoms can be explained by anemia. Yes, red blood cell isolated from patient show abnormally low level of lactate production. Very good, you have given the correct answer, low level of lactate production. If yes, deficiency of which of the following enzyme would be most likely to cause? Yes, answer is pyruvate kinase deficiency. Very good. So in if pyruvate kinase is deficient, then pyruvate will not be formed, then lactate will not be formed. Yes, so lactate would be low, lactate would be low. Pyruvate kinase deficiency can lead to low lactate. Now in the same question, if I change high lactate level, then yes, the answer would become yes, high lactate level. Now look at the screen. I told you, yes, glucose is normally diverted towards HMP shunt, also diverted toward glycolysis. Yes, if HMP shunt G6P deficiency, glucose 6 phosphate dehydrogenase is the enzyme of HMP shunt. If this step will not occur, more and more glucose will enter glycolysis. Again I say, yes, again I say, and I will discuss shortly HMP shunt. In HMP shunt, I will tell you that in HMP shunt, pentose phosphate pathway, glucose 6 phosphate is converted to 6 phosphogluconate by glucose 6 phosphate dehydrogenase. If this enzyme is deficient, then glucose 6 phosphate will not enter HMP shunt. More and more glucose 6 phosphate will enter glycolysis. More and more glucose phosphate will enter glycolysis. So more and more lactate will be produced. That's why lactate level would be high, glucose 6 phosphate dehydrogenase. Very good. If high, the answer is this. In the same question, if I say there is exercise intolerance, same question means low lactate level and exercise intolerance. Low lactate level and exercise intolerance, it is type 7 glycogen storage disease. I will discuss in detail. For the time being, type 7 glycogen storage disease, that is Terrois disease. Terrois disease. So Terrois disease, yes, in this phosphofructokinase is deficient. Phosphofructokinase is deficient. This deficiency occur in RBC, and this deficiency also occur in muscle. RBC and muscle. So if it occur in RBC and muscle, yes, phosphofructokinase deficient. In RBC there would be hemolysis. In muscle, if phosphofructokinase is deficient, then energy will not be produced, but there would be exercise intolerance. So that same question answer would become phosphofructokinase. Means if low lactate level and exercise intolerance, the answer would become phosphofructokinase. Same question, 3 answers. So now in the same question, if I remove this line, did you understand this? First of all, let me tell you, if this phosphofructokinase deficient in RBC, hemolysis, muscle, exercise intolerance. If phosphofructokinase is deficient in muscle, you can do exercise, that is called exercise intolerance. Clear? Now if I remove this, what is the answer? Please do reply. In this means they have not mentioned anything, they are asking which hemolytic anemia is this. Yes, you will prefer to answer D. So most common is this. Okay, why answer is D? Because if a patient comes to me with hemolytic anemia, the first diagnosis which will come in my mind would be G6 deficiency. Second differential diagnosis would be pyruvate kinase. Third point would be, yes, that may be phosphatidylkinase. So you understood what is the scenario and why we asked mark question. Yes, so every time read question carefully. Okay.
Hi friends, so like, share and subscribe. If you have any doubt, comment below. Thank you.